Hello. In this video, we will cover the many plot options for omics data in mode, and how to make each type of plot. There are 8 plot types in mode, with slight differences between RNA-Seq and mass spectrometry or nuclear magnetic resonance data. As a reminder, in Trelloscope, the term panel refers to a single plot. Here, the terms panel and plot are used interchangeably. So when we say the term paneling by, we mean that a plot will be generated for each panel. If that panel is a peptide, there will be one plot per peptide. If the panel is a sample, there will be one plot per sample, and so on. There are 4 types if paneling by biomolecule. For MS or NMR data, if paneling by the biomolecule and plotting abundance, the options are abundance box plots, which will be split by group if sample information is provided, and abundance histograms. If paneling by biomolecule and plotting missingness, the option is a missingness bar plot, which will also be split by group if that information is provided. If paneling by biomolecule and plotting fold changes, fold change bar plots are available. These plots will give the fold change per comparison. Significant fold changes will have larger borders. There are 2 types of plots if paneling by sample. If paneling by a sample, abundance box plots and missingness bar plots are available. There are 4 plot types if paneling by a biomolecule information column, like the protein each peptide belongs to. If paneling by biomolecule information and plotting abundances, abundance heat maps are available. Note that in this case, z-scores for each abundance are plotted. If grouping information is provided, the y-axis, which is the samples, will be sorted by group. If paneling by biomolecule information and plotting fold changes, there are fold change box plots, fold change heat maps, and fold change volcano plots. All fold change plots have the capability to indicate significance. Fold change volcano plots can be filtered down to a specific comparison, though the default is to build plots for all comparisons. Now, if users have RNA-Seq data, the fold change plots will remain the same. Instead of abundances, log counts per million are calculated in RNA-Seq box plots, RNA-Seq histograms, and RNA-Seq heat maps. Instead of missingness, non-zero count bar plots are made. Now that we know the plotting options available in mode, let's learn how to make them. First, select your panel by choice, which will be either a biomolecule, a sample, or a column from the biomolecule information file. These panels are a Trelloscope term that is synonymous with plot, meaning that one plot will be generated per panel, biomolecule, sample, or biomolecule information column. Next, select which data to plot, which is abundance, missingness, or fold change for MS or NMR, or for RNA-Seq, non-zero, fold changes, and RNA-Seq values. Next, you may select a panel variable for building your plot, but it is not required and does not change the results. Now, click a row on the table generated in the Select plot to view different plots. Click Confirm Selection to pick this plot. If a mistake was made, click Make Another Selection. Next, you may modify the plot's labels, font sizes, tick angles, tick font sizes, titles, title sizes, legends, color schemes, etc., and widgets below the plot. Other options include the ability to flip the X and Y axis, as well as remove the legend. When done making changes to the plot, click Redraw Plot and see if you are happy with the results. Note that the text is smaller in this view of the plots than it will be in the final Trelloscope due to differences in how internet browsers render apps and how Trelloscope displays settings. Next, if you would like the display to build faster, you can filter them down in the optional Filter Plot step. Check out the Filter Plot step video next for more information. If you're not interested in this, skip that video and go to the Create Trelloscope step.